Good afternoon, all of you. Now we are going to discuss another important step in RDNA technology, that is selection of transformed cells. Selection of transformed cells from the non-transformed cells. Okay, what is transformation process? What is transformation principle or transformation process here? Right. So transformation is a procedure by which introduction of foreign genetic material into host cell cytoplasm called transformation. So definition for transformation. So transformation is a procedure by which introduction of rDNA, otherwise introduction of foreign DNA into host cell cytoplasm called transformation process. But in this rDNA technology, so we need to select transformed cells from the non-transformed cells. Why? Because so we need only transformed cells. We don't need non-transformed cells. We need to separate non-transformed cells from the transformed cells. For example, you can take PBR322 vector, artificial vector. So that PBR322 artificial vector has antibiotic resistant genes like ampicillin gene, tetracycline resistant gene. Right, it means that vector has ampicillin resistant gene and tetracycline resistant gene. Okay, right. If we will insert foreign genetic material or other organism genetic material into PBR322 vector, PBR322 vector, it resulting in the formation of rDNA or DNA. Okay, right. So when we'll insert that rDNA into host cell cytoplasm, for example, you can take E. coli cell, act as host cell. Okay, when we will insert PBR322 rDNA molecule into E. coli cell cytoplasm, so that E. coli cell exhibits resistant to antibiotics like ampicillin and tetracycline. Ampicillin and tetracycline. It means that Escherichia coli bacteria transformed to the transformed to the antibiotic resistant genes. In general, that Escherichia coli bacteria do not carry antibiotic resistant genes. But when we will insert PBR322 rDNA molecule into E. coli cell cytoplasm, so that E. coli cell exhibits resistant to antibiotics like ampicillin and tetracycline. Right. So now come to sixth important step, how we will select transformed cells from the non-transformed cells. We will see step by step, right? So for example, you can take PBR322 vector. Which vector? PBR322 vector. Yes, imagine it is vector. PBR322 vector. See, it has ampicillin resistant gene, AMPR, ampicillin resistant gene. And this segment indicates that TETR, tetracycline resistant gene. Tetracycline resistant gene and ampicillin resistant gene. Okay, right. In tetracycline resistant gene, we observed two palindromic sequences or cloning sites. What are they? BAMH1 sequence and another one is SAL1 sequence, right? So in PBR322 vector, so the tetracycline resistant gene has two important cloning sites. One is BAMH1 sequence and another one is SAL1 sequence. Okay, right? So we selected foreign genetic material or alien DNA. Better to take it as foreign DNA or desirable DNA. So better to write it as desirable, right? Either desirable DNA or foreign DNA or alien DNA, same meaning, right? So we need to insert this desirable DNA into the PBR322 vector. Right. So what type of restriction endonuclease enzymes we are using for cutting foreign genetic material and PBR322 vector here? Right. So BAMH1, BAM1, small, simple form it is BAM1, right? BAMH1. restriction endonuclease enzyme we are using for cutting of desired genetic material and vector genetic material. So when we are using BAMH1 restriction endonuclease enzyme, it resulting in the formation of right sticky ends. Where exactly? At BAMH1. BAMH1. So the desirable DNA and BAMH1 sequence, right, fragmented by the restriction endonuclease enzyme. Which restriction endonuclease enzyme here? So BAMH1, right? So BAMH1 restriction endonuclease enzyme resulting in the formation of sticky ends. Sticky ends, right? See like this. Sticky ends, right? Here also same. Sticky ends. See like this. Sticky ends. Right. By using ligase enzyme, we have to add the desirable DNA. We have to insert the desirable DNA into the PBR322 by using ligase enzyme. So we inserted the desirable DNA into the vector. It resulting in the formation of our DNA molecule now. But in this process, so two types of vectors are formed. Two types of vectors are formed. So one is altered vector and another one is unaltered vectors. See, some vectors are altered, some vectors are unaltered. For example, see here, this vector has, this PBR322 vector has inset. Inset means foreign genetic material. We call it as altered or unaltered vector. Altered vector. Altered vector. Right. Then, but some vectors do not have any inset. Some vectors do not have any inset. So we call them as unaltered vector. Unaltered vectors. Unaltered vectors and altered vectors, right? So these two types of vectors are formed, altered vectors and unaltered vectors. So these two types of vectors again transferred into the host cell cytoplasm. So but here we are taking only one PBR322 vector now, sir. Yes, so it is not possible to insert only one foreign segment into the one vector. It is not possible to insert only one foreign genetic material into the one vector. Why? Because at a time, we need to collect many plasmids, otherwise many PBR322 vectors in one test tube, right? So many foreign DNA fragments in another test tube. For example, so in one test tube, we collected many plasmids. Which type of plasmids? PBR322 plasmids, artificial plasmids in one test tube. In another test tube, we collected foreign DNA segments. Okay, right? So what type of enzyme we need to add in the test tubes? Okay, right? BAMH1 restriction endonuclease enzyme we need to add into the plasmids and foreign DNA segments. So it resulting in the formation of sticky ends as usual. Sticky ends. But here, two types of vectors are formed. Why? Because some vectors having insets. We call them as altered vectors. But some vectors so do not have any inset. It means so that DNA, foreign genetic material, not inserted into the some vectors, right? So we call them as unaltered vectors. So at a time, two types of vectors are formed. Don't confuse here. In one test tube, we collected PBR322 vectors. For example, 100 PBR322 vectors we collected in one test tube. In another test tube, approximately 50 foreign genetic material or 50 foreign DNA segments we collected in another test tube, right? So both of them treated with restriction endonuclease enzyme. Which enzyme? BAMH1 sequence. BAMH1 enzyme, right? That enzyme resulting in the formation of stick ends, right? So again, we have to add that foreign segments into the plasmid test tube by ligase enzyme. Again, we have to add ligase enzyme also. So with the help of ligase enzyme, that foreign genetic materials inserted into the vectors. But maybe foreign genetic material segments are less, right? So what PBR322 vectors are more. In that case, so some vectors have insets. We call them as altered vectors. Some vectors have some vectors do not have any inset. We call them as unaltered vectors. See, whenever foreign genetic material inserted at the place of BMH1, the tetracycline resistant gene completely inactivated. Is it clear? Whenever desirable genetic material inserted at BAMH1 sequence, the tetracycline resistant gene inactivated. So now come to here. How many types of vectors are formed here? Two types, altered and unaltered vector. So now tell me, altered vector resistant to only ampicillin. Altered vectors resistant to only ampicillin antibiotic. Sir, why not tetracycline? Sir? Why not tetracycline? Why? Because of insertion of foreign DNA, the tetracycline resistant gene inactivated. But here, that unaltered vectors resistant to both antibiotics, ampicillin and tetracycline. Ampicillin and tetracycline. Why? Because there is no insert. There is no foreign genetic material. It is an unaltered vector. Right. So unaltered vectors resistant to both ampicillin and tetracycline, but altered vectors are resistant to only ampicillin, but not to tetracycline. Why? Because at the place of tetracycline resistant gene, foreign genetic material inserted. Right. Then, so these two types of vectors, altered and unaltered vectors, transferred to the host cells. Transferred to the host cells, right? So these three types of these two types of vectors transferred to the host cells. Transferred to the host cells. See here, it is one bacterial cell. One type of bacterial cell. This is another type, right? It is one more type of bacterial cell, right? See, when we insert the two types of vectors into the host cell cytoplasm, so some bacterial cells receives altered vectors. Some bacterial cells receives altered vectors. So we called it as transformed cell with altered vector or unaltered vector. Transformed cell with altered vector. Right? Then come to here, second bacteria. In second type of bacteria has unaltered vector inserted into the second type of bacteria. So we call it as transformed cell with altered or unaltered vector, unaltered vectors. Right. Then what about third type of bacteria? So do not have any plasmid, either it is transformed or non-transformed, whatever it may be. So here altered vector, unaltered vector, both of them absent in third type of bacteria. So we call it as non-transformed cell. Is it clear? So like that, three types of bacterial cells formed, right, when we insert altered and unaltered vectors into host cell cytoplasm, right? So these three types of bacterial cells, again, we transferred into the ampicillin medium. Which medium? Ampicillin medium. For example, so you can take Petri plate or Petri dish. It is Petri plate or Petri dish. Okay, we transport these three types of bacterial cells into the Petri plate, which contains ampicillin medium, right? So this Petri plate has ampicillin plus nutrient medium. Nutrient medium. So in this nutrient medium and ampicillin medium, which bacterial cells can survive and which bacterial cells can eliminate, right? So now tell me, in this case, in this medium, which type of bacterial cells can survive and which type of bacterial cells eliminates, right? See, first type of bacterial cell can survive. Why? Because it has ampicillin resistant gene, right? So it has inside altered vector it is, but it is resistant to ampicillin antibiotic. Come to second cell again. So that second cell has unaltered vector. Unaltered vector means it is both for ampicillin and tetracycline resistant capacity, right? So this vector or this transformed cell resistant to antibiotics, both antibiotics, ampicillin and tetracycline. But come to third one, it means second one also survives. Second type of bacteria also survives. But what about third one, sir? Non-transformed cell. So that non-transformed cell leads to death, right? So that non-transformed cells completely eliminated from the master plate. We call it as master plate. So in that master plate, which type of medium we observed? Ampicillin medium. So in that ampicillin medium, only transformed cells can survive and non-transformed cells eliminates. Right? It means what is the use of antibiotic resistant gene ampicillin here? So that ampicillin resistant gene helps in separation of transformed cells and non-transformed cells, right? So it means it helps in identification of non-transformed cells and elimination of non-transformed cells, right? So what is the use of ampicillin resistant gene here? So identification and elimination of non-transformed cells from the transformed cells. Is it clear? But what type of bacterial cells we required here? Right. So transformed cells with altered vector. Transformed cells with altered vector, right? We required. But what about other type of bacterial cell? Transformed cells with unaltered vector. So the transformed cells with unaltered vectors, right? We not required, right? We don't want transformed cells with unaltered vectors in RDNA technology, right? We require transformed cells with altered vectors. But how we will separate transformed cells with altered vector from the unaltered vectors, right? So come to here again. Next, so the two types of bacterial cells transfer to another plate, another Petri plate. So we call it as culture plates. Culture plates. So what type of medium we observed in culture plates? Yes, that is tetracycline antibiotic. Tetracycline antibiotic plus nutrient medium. Nutrient medium. Right. So by using velvet cloths, when we press that velvet cloth on the ampicillin medium, so these two types of transformed cells, right, transferred to the velvet cloth. So again, when we'll press that velvet cloth on the tetracycline medium, the two types of bacterial cells also transferred to culture plates. Culture plates. Right? So it is main genetic material and it is altered vector. So altered vector bacterial cell it is. Then this is unaltered vector bacterial cell. So above one is altered vector bacterial cell. Below one is unaltered vector bacterial cell. So now tell me, these two types of bacterial cells we transfer to the culture plate which contains tetracycline nutrient medium. Right. So in this Petri plate, what type of bacterial cells can survive? What type of bacterial cells can eliminate? Eliminates. Come to here. See, first one. It has inset now at the place of tetracycline resistant gene. It has inset. It means so the tetracycline resistant capacity completely decreased in first type of bacterial cell. So that is transformed cells with an altered vector. Altered vector. So completely these bacterial cells eliminates from the culture plates. Culture plates. Then in second condition. So transformed cell with unaltered vector. So the transformed cells with unaltered vector bacterial cells can survive in tetracycline nutrient medium. Why? Because it has both tetracycline resistant gene and ampicillin resistant gene. Is it clear? But how? We need first type of bacterial cells only. But the bacterial cells completely leads to death in tetracycline nutrient medium. Right? But how we will identify some? How we will identify some? We need this type of bacterial cells only. But in culture plate, so that bacterial cells completely leads to death due to the inactivation of tetracycline resistant gene. But in this Petri plate or master plate, we observed both transformed cell with unaltered vector, transformed cells with altered vectors. But we require altered vector containing bacterial cells only. Right? So don't confuse here. See, three types of bacterial cells we transfer to the master plate. So in that master plate, which antibiotic is there? Ampicillin. Right? So here two are transformed cells. Why we call it? Why we call them as transformed cells? Right? Other genetic material PBR322 and foreign genetic material, right, enters into host cell cytoplasm now here. Other genetic material. That's the reason both of them are called transformed cells. But here there is no PBR322 and foreign DNA. All right, we call it a non-transformed cells. So there is no antibiotic resistant genes also here in this bacterial cell. Right, we call it a non-transformed. These two are transformed. See that two transformed cells are resistant to antibiotic ampicillin. So this side, ma. So this side ampicillin resistant genes are there now in plasmid. Yes. So both of them exhibits resistant power to the ampicillin antibiotic. But what about third one? So there is no resistant genes to the antibiotic ampicillin, right? So that's the reason non-transformed cells eliminates from the transformed cells here. Right. Then, so we need first type of bacteria only, altered vector containing bacterial cell. Both of them are transformed cells, but we need altered vector containing bacterial cell. Right? We don't want unaltered vector containing bacterial cell. But how we will isolate so? How we will separate so? Right? And that one more thing here. So altered vector containing bacterial cell is called a recombinant cell. A recombinant cell. Unaltered vector containing bacterial cell is called non-recombinant cell. So don't confuse transformed cells, non-transformed cells. Recombinant cells, non-recombinant cells. So here both cells are considered as transformed cells. This cell is considered as non-transformed cell. Right? Non-transformed cells completely eliminated here. But in this case, in these two transformed cells, we need to select a recombinant cell and non-recombinant cell. Right? So this is a recombinant cell. Simple term, recombinant cell. And it is non-recombinant cell. Why? Because it has inset or foreign genetic material, desirable genetic material. So we call it as recombinant cell, non-recombinant cell. We need recombinant cell only. We require recombinant cell only. Right? That's the reason we transferred that recombinant and non-recombinant cells to the culture plate. Culture plate. So in this culture plate, due to inactivation of tetracycline resistant gene, so the bacterial cell leads to death. Which bacterial cell? Recombinant bacterial cell. We need recombinant cell only, but that recombinant cell leads to death. Then second one, non-recombinant cell can survive in tetracycline medium, but it is not useful. It is not useful. But how we will isolate? So how we will separate desirable, recombinant bacterial cells? So, right? It is possible when we observe both culture plate and master plate at a time. When we observe under microscope, right? It is possible to identify, right, recombinant cells from the non-recombinant cells. Recombinant cells from the non-recombinant cells. But how so? How so? See, in culture plates, the bacterial cells which cannot survive in culture plates and the bacterial cells which can survive in master plate, those bacterial cells only we required. But how we will identify, sir? How we will identify, sir? So what is which bacteria leads to death in culture plate, which bacterial cells can survive in master plate? How we will identify, sir? See, in earlier classes already I told you one thing. When we insert other organism genetic material into bacterial cell cytoplasm or any organism, so slowly some phenotypical characters or external characters leads to change. Is it clear? So when we insert other organism genetic material, so some phenotypical characters leads to change. So here also same thing, that recombinant, right? So that recombinant DNA containing bacterial cells exhibits some special characters, right? So now come to here. So the bacterial cells which leads to death in culture plate and which can survive in master plate, so those bacterial cells are considered as recombinant cells, and they separated the recombinant cells from the master plate. So when it is possible, when we observe both Petri plates at a time under microscope, it is possible. It is possible, right? And second case, the bacterial cells which can survive in culture plates, the bacterial cells which can survive in culture plate and master plate, not required for RDNA technology. Those are considered as non-recombinant cells. Non-recombinant cells. Is it clear? So the bacterial cells which can survive in both culture plate and master plates are considered as non-recombinant cells. That non-recombinant cells not required for RDNA technology, RDNA process, right? So easily we will separate that bacterial cells which can survive in both culture plate and master plate. Easily we will separate, right? And the bacterial cells which can survive only in master plate, not in culture plate, so those bacterial cells only considered as recombinant cells, and those recombinant cells separated from the master plate and used in RDNA technology, right? So in this way, recombinant cells selected from the non-recombinant cells, transformed cells selected from the non-transformed cells, right? Done. So just you have to draw that schematic representation, that information on it. Just you have to draw that schematic representation. Easy technique and simple technique. So we are using three methods for the selection of transformed cells from the non-transformed cells. In that, first one is by using selectable markers. By using selectable markers, small heading you have to write here. For the selection of transformed cells from the non-transformed cells, we are using three methods or three techniques. In that, first one is by using selectable markers. By using selectable markers. So what are selectable markers here? Selectable markers are nothing but antibiotic resistant genes. Antibiotic resistant genes. Right. So again, I'm repeating. Don't confuse. See, in process of RDNA technology, we selected PBR322 vectors. At a time, we collected PBR322 vectors in one test tube and foreign genetic material in another test tube. Right? Then, second step. See, both of them, PBR322 and foreign genetic material, treated with restriction endonuclease enzyme. Which enzyme? BAMH1 restriction endonuclease enzyme. So it resulting in the formation of sticky ends, same type of sticky ends. Again, after that, we transferred that foreign DNA molecules from the one test tube, right, into plasmid containing test tube. All right, simply we have to transfer foreign DNA segments into the plasmid test tube, right? After that, we have to add ligase enzyme. So it resulting in the formation of two types of vectors. So some vectors having insert or foreign genetic material, we call it as altered vector, right? Some vectors do not have any inset, we call them as unaltered vectors. So that altered vectors exhibits resistant power to the ampicillin only. Why? Because ampicillin resistant gene is present in altered vector, but tetracycline resistant gene inactivated. Why it is inactivated? Why? Because it has inset at the place of tetracycline resistant gene. Is it clear? At the place of tetracycline resistant gene, it has that altered vector has desirable DNA. Right. Come to here. In unaltered vectors, resistant for both ampicillin and tetracycline. Why? Because they don't have any inset or foreign DNA, desirable DNA. Right. So these two types of vectors transferred to the inserted into the host cells. Transferred to the host cells or inserted into host cells, right? So in this case, three types of bacterial cells are formed. So some bacterial cells having altered vectors, some other bacterial cells have unaltered vectors, some other bacterial cells do not have any vectors, right? So we call them as transformed cells and non-transformed cells. So the bacterial cells which have PBR322 vectors, either it is altered or unaltered, whatever it may be, called transformed cells, right? So the bacterial cells which do not have that PBR322 called non-transformed cells. Non-transformed cells. Is it clear? So what are transformed cells? What are non-transformed cells? Already I told you definition. Transformation is a procedure by which insertion of foreign DNA or other DNA into host cell cytoplasm called transformation procedure. See here, it is also transformed cell. Why? Because PBR322 and desirable DNA inserted into host cell cytoplasm. In this case also, only PBR322 enters into cytoplasm. Both of them are transformed cells, but it is unaltered vector containing transformed cell. It is, sorry, altered vector containing bacterial cell, but it is unaltered vector containing bacterial cell, but both are transformed cells. But third one is non-transformed cell. There is no any type of vector or foreign DNA, right? So these three types of bacterial cells we transferred into the master plate, which contains ampicillin nutrient medium. Ampicillin nutrient medium. Okay, right? So what type of bacterial cells can survive in master plate here? Right, the bacterial cells which have ampicillin antibiotic resistant genes can survive. The bacterial cells which lacks ampicillin resistant genes eliminates. It means non-transformed cells completely eliminates from the medium. Transformed cells can survive. All right. Why? Because both transformed cells exhibits ampicillin resistance capacity. Right. Then. So, but we need recombinant cells in the transformed cells. Both of them are transformed cells, but we need only recombinant cells, right? So how we will separate recombinant cells from the non-recombinant cells, right? Both of them are transformed cells. Altered vector containing bacterial cell is considered as recombinant cell. Unaltered vector containing bacterial cell is called non-recombinant cell. We required only recombinant cells, right? So how we will separate? So see, simultaneously, these two plates, when we'll observe under microscope, easily we will separate. How so? See, these two types of bacterial cells by using velvet cloth transferred into culture plate, which contains tetracycline medium. In the tetracycline medium, so recombinant cells completely eliminated due to inactivation of tetracycline resistant gene, right? So due to insertion of foreign DNA at the place of tetracycline resistant gene, it is completely inactivated. So it leads to death in tetracycline medium. So which bacterial cells can survive in culture plate? Only unaltered vector containing cells. Why? Because that unaltered vector has both ampicillin, tetracycline resistant genes, right? So these two plates at a time we observe under microscope. The two types of bacterial cells, two plates bacterial cells at a time observed under microscope, right? Okay, then based on the phenotypical characters, easily will identify what are recombinants, what are non-recombinants, right? So these are recombinants. So the bacterial cells which leads to death in culture plate, which can survive in master plate or recombinant cells. Right? So that cells which are absent in culture, easily the cells which are absent in culture plates, the cells which are present in master plate are recombinant cells. Right? So that type of cells only we collected for next procedure. And the bacterial cells which are present in both culture plate and master plate not required for further procedure or further process of RNA technology. Right? So in this way, we collected recombinant cells from the non-recombinant cells. We selected recombinant cells from the non-recombinant cells, and that too we collected transformed cells from the non-transformed cells. Right? So we selected transformed cells from the non-transformed cells, and finally we have to collect recombinant cells from the non-recombinant cells. Right? Now tell me, which type of resistant genes helps in selection of transformed cell from the non-transformed cell? Right? So only two questions here. Which type of antibiotic resistant gene or selectable marker helps in identification of transformed cells from the non-transformed cell? Ampicillin resistant gene. It means one selectable marker, ampicillin resistant gene, helps in identification of transformed cells from the non-transformed cell. And second one, another selectable marker, that is tetracycline resistant gene, inactivated due to insect, but it helps in selection of recombinant cells from the non-recombinant cells. Right? So finally, conclusion here is, so that the two types of antibiotic resistant genes which are present in PBR322 vector helps in selection of transformed cells from the non-transformed cells, selection of recombinant cells from the non-recombinant cells. That's the reason that antibiotic resistant genes are considered as selectable markers. Selectable markers. Why we called antibiotic resistant genes as selectable markers? For the selection of transformed cells from the non-transformed cells, for the selection of recombinant cells from the non-recombinant cells. That's the reason we call them as selectable markers. Is it clear? So which type of selectable marker helps in identification of transformed cells from the non-transformed cells here? Ampicillin resistant gene. And which type of selectable marker, right, helps in identification of recombinant cells from the non-recombinant cells? Tetracycline resistant gene. Right? So just you can take on screenshot now. Total information I given here, diagrammatically I explained here. Right? So better to take this schematic representation for that, for the selection of transformed cells from the non-transformed cells by using selectable markers. But it is hard procedure, cumbersome procedure or hard procedure for the selection of transformed cells from the non-transformed cells. Cumbersome procedure, it means hard procedure. Hard procedure. All right. Then second method again. Second method. Why it is difficult? Why it is cumbersome procedure? Why it is hard procedure? Why? Because so we need to observe both plates at a time under microscope, right? We need to observe, right? We need to place that both plates at a time. It is hard procedure or cumbersome procedure, right? Right. That's the reason scientists developed another technique for the selection of recombinant cells from the non-recombinant cells, that is insertional inactivation method. Insertional inactivation method. Second method. Right. For the selection of transformed cells from the non-transformed cells, or for the selection of recombinant cells from the non-recombinant cells. So we are using another method or second method, that is insertional inactivation. Insertional inactivation. Second method, insertional inactivation. So in this method, based on the ability to produce color, easily we will identify a recombinant cells from the non-recombinant cells, right? So in insertional inactivation method, so that the recombinant cells easily will identify from the non-recombinant cells based on the color, based on the ability to produce color. So that bacterial cells, recombinant bacterial cells separated from the non-recombinant bacterial cells in this process. But here we're using PUC19 or PUC101 vectors. PUC19 or PUC101 vector. So better to take PUC19 vector here. 19 vector. As usual, that PUC19 vector also has both antibiotic resistant genes, ampicillin and tetracycline resistant gene. But in addition to these two antibiotic resistant genes, it has another sequence that is B-Gal sequence, beta galactosidase enzyme sequence. So that beta galactosidase enzyme sequence located in PUC19. PUC19. So what is the use of beta galactosidase enzyme sequence here? That beta galactosidase enzyme sequence, right, responsible for the synthesis of beta galactosidase enzyme. Beta galactosidase enzyme. So that B-Gal sequence, which is located in PUC19, right, undergoes transcription and translation and synthesizes beta galactosidase enzyme. But already we have some idea, right, that beta galactosidase enzyme helps in conversion of lactose into glucose and galactose, right? So that beta galactosidase enzyme, right, encoding gene, that enzyme encoding gene located in PUC19. PUC19. Okay, right. Then imagine it is foreign genetic material. Foreign genetic material, otherwise desirable DNA, desirable genetic material. So we need to insert the desirable genetic material at the place of B-Gal sequence, beta galactosidase enzyme sequence. Is it clear? See, we need to insert foreign genetic material or alien DNA or desirable DNA into the PUC19 vector at the place of B-Gal sequence. B-Gal sequence. So when we insert the desirable DNA at the place of B-Gal sequence, so that beta galactosidase enzyme sequence completely inactivated. So we call it as insertional inactivation. Insertional inactivation. Is it clear? So whenever desirable DNA inserted into the PUC19 vector at the place of B-Gal sequence, so that beta galactosidase enzyme sequence completely inactivated, we call it as insertional inactivation method, right? Then see, at a time we collected many PUC19 vectors in one test tube, many foreign DNA segments in another test tube, both of them treated with same type of restriction, endonuclease enzyme like selectable markers, right? And they're resulting in the form of same type of sticky ends. By using ligase enzyme, we inserted foreign genetic material into vectors. Vectors, they're resulting in the formation of two types of vectors again. Here also, they forms two types of vectors. What are they? One is altered vector and another one is unaltered vectors, right? So one is altered vector. So here foreign genetic material inserted. in this vector foreign genetic material inserted. Is it altered or unaltered? Altered. Altered vector. Then come to this side. It is unaltered vector. Unaltered vector, right? Unaltered. Unaltered vectors. As usual, same thing, both of them, altered and unaltered vectors transferred to the host cells. So when we transfer that unaltered and altered vectors into the host cell cytoplasm, three types of bacterial cells are formed again. Three types of bacterial cells, right? So this is transformed cell with altered or unaltered, transformed cell with altered vector. Altered vector. Then second one, transformed cell with unaltered vector. And third one is transformed cell, sorry, non-transformed cell it is. Second one is transformed cell with unaltered vector. Unaltered vector. And last one is non-transformed. Non-transformed. Right? As usual, the three types of bacterial cells again transferred to the amphicoelin medium. But how we will select recombinant cells from the non-recombinant cells in this process or in this insertional inactivation method? So we'll see in next class. In detail, we'll see in next class.